Good day, ladies and gentlemen. I'm delighted to welcome you to this online seminar and live panel, Immunotherapies, New Perspectives for the Treatment of Cancer Patients. During this seminar, we will demonstrate what the enormous opportunities are that lie ahead to generate breakthrough treatments in cancer. Innovative immunotherapies are becoming more and more a reality and will hopefully bring a better future for a bigger and bigger number of cancer patients. To give us an insight on the trends, the challenges and the future in this field, I'm honored to welcome an experienced panel of experts today. We will exchange with Professor Sophie Lucas from the Belgian de Duve Institute, a multidisciplinary research institute that is a part of the UCL University. Later on, Dr. Halioua, the CEO of the Belgo-French biotech company PDC-Line Pharma, will present the latest results in the development of innovative cancer vaccines, which it hopes to bring to the market in the coming years. His colleague, Dr. Joël Plumas, CSO of PDC-Line Pharma, and Dr. Javier Carrasco, a clinical oncologist from the Hôpital de Charleroi in Belgium, will discuss how an innovative technical approach, the quantitative analysis of T lymphocytes receptor repertoires, is proving instrumental to expedite the development of the company's cancer vaccine pipeline. We hope to bring you new insights and inspire you about all these developments. Throughout this seminar, you can ask questions via the chat box on your screen. And after the three presentations, we will hold a Q&A session with all of our experts. Thanks to all of you for joining us today. So now let's kick off this seminar without further ado. Welcome, Professor Sophie Lucas. Can you please introduce yourself to us?
Sure, thank you. So I was trained as an MD at the University of Louvain, and then I obtained a PhD degree for work performed in the field of tumor immunology, where we discovered novel tumor-specific antigens that can be used as cancer vaccines. Then I continued my training as an immunologist by doing a postdoc at Genentech in California, and there I was working on cytokines and cytokine receptors. And then I came back to Belgium and founded my own research group at the de Duve Institute, still on the medical campus of the UCLouvain. And there I had a group of about 15 scientists working in the field of immunosuppression and tumor immunology. I'm also professor of cancer immunotherapy at the UCLouvain.
And what is your research group focusing on today?
So we are interested in determining how regulatory T cells, called Tregs, can suppress immune responses, particularly in the context of cancer. We've identified a mechanism that implies expression of GARP, a protein on the surface of Tregs, which helps them produce a very potent immunosuppressive cytokine named TGF-beta, and we've developed an approach to block TGF-beta production by GARP-expressing Tregs. This approach corresponds to monoclonal antibodies, which are currently being tested in phase 1 clinical trials as novel approaches for cancer immunotherapy.
This new field is called immuno-oncology. Can you give us a definition of it?
Yes, sure. Immuno-oncology is a branch of oncology which resorts to therapeutic approach that aim at pharmacologically targeting the immune system, immune cells or molecules expressed on immune cells to increase the immune responses that patients can mount against their own tumor. So these cancer immunotherapy approaches are actually what defines the field of immuno-oncology.
This field has been on the rise for quite some time now. Can you explain to us why more and more attention and investment is being paid to it?
Well, I guess that this is because there have been amazing developments in the last 10, 15 years, let's say, that were made thanks to two major steps in fundamental immunology, which were made thanks to discoveries that arose in the last 30 years. The first of this very important turning point was actually the identification by Thierry Bohn and his colleagues working at the de Duve Institute of so-called tumor antigens that are recognized on the surface of tumor cells by lymphocytes able to recognize these antigens thanks to the expression of an antigenic receptor called the TCR, T-cell receptor, which is unique to each clone of anti-tumor T lymphocyte and which can recognize these antigens on the presence of tumor on the surface of tumor cells. Tumor antigens can be, and this is what Thierry Beaune has demonstrated, strictly limited to the surface of tumor cells. That is, they can be expressed only on tumor cells and not on normal cells of the organisms. These tumor antigens are called tumor-specific antigens, and this explains why T lymphocytes directed against these antigens can induce immune responses that eliminate the tumor cells without causing harm to normal cells. The discovery of these tumor-specific antigen opened the way to the development of a very specific form of cancer immunotherapy, which implies the use of these antigens as cancer vaccines. A second major step was done a little bit later, at the end of the 90s, beginning of the 2000s, by these two persons, which are represented here, Jim Allison and Tasuku Onjo. And they each independently discovered molecular mechanisms that explain why anti-tumor T lymphocytes are actually hampered in their function to eliminate tumor cells in many cancer patients. They discovered receptors present on the surface of anti-tumor T lymphocytes which inhibit the activity of these lymphocytes. They called them, and I think it's Jim Allison who called these receptors the immune checkpoints, and following up on their discoveries, both Jim Allison and Tasuku Onjo developed monoclonal antibodies to block these inhibitory receptors, and these monoclonal antibodies are often called immune checkpoint inhibitors. This is why they both obtained the Nobel Prize in Medicine in 2018, because these discoveries actually led to the development of novel therapeutic approaches which really drastically changed the face of oncology and defined somehow the successes of current immuno-oncology.
It will drastically change oncology, you say? Can we predict the potential of immunotherapeutics?
Yes, so I think the potential is huge because already the successes that have been observed in the last 10-15 years have been really important, but they are probably going to continue progressing because there is a lot of room for improvement, discovering many other immunosuppressive mechanisms that can also be pharmacologically targeted, for example, or also discovering many antigens that can be used as cancer vaccines. There is a lot of room also for personalized medicine with the identification of personal antigens corresponding to mutated antigens which are present only in one patient and one tumor type. And as I said, also many immunosuppressive mechanisms, so some of which will be active in some types of tumors or patients and not in others.
Personalized medicine is the way forward. What are the different medical approaches that are being researched currently?
So actually, all can be summarized also in this very simplified scheme, right? Where you have the representation of the tumor antigens, the anti-tumor lymphocytes, which are the effectors of the immune responses, and then the immunosuppressive molecules. When you use the tumor antigens themselves to administer them in cancer patients in a very immunogenic fashion, you do so-called vaccination, trying to reinvigorate or stimulate a new anti-tumor T lymphocyte that will be directed very specifically against the tumor cells and not the normal cells if you use tumor-specific antigens. So the cancer vaccines have not been tremendously successful until now because of the lack of immunogenicity of the vaccines that have been used today. But I think that this will change really soon thanks to combinations with other immunotherapeutic approaches.
And we will have more about that later on this afternoon.
Absolutely, I'm sure. And the second approach is an approach based on cellular therapy. It consists in transferring into cancer patients lymphocytes that's directed against tumor cells that have been amplified in vitro or even genetically manipulated to express receptors that are specific of antigens present on the tumor cells. And the examples of such cellular therapies, the most famous examples correspond to the CAR T cells, for example, which express chimeric antigenic receptor on the surface of lymphocytes and that are being used currently in hematological malignancies, mainly, not yet a lot in solid tumors. This is cellular therapy, so it's very heavy, very costly, but it has given some interesting, very interesting results in hemopathies. And then finally, you have the famous immune checkpoint blockers, these immunostimulatory antibodies that interrupt the CTLA-4/B7 axis, inhibitory axis, for example, so antibodies against CTLA-4 developed by Jim Allison first, and the antibodies that interrupt the PD-1, PD-L1 inhibitory axis directed against either PD-1 or its ligand, PD-L1, and that were developed by Tasuku Honjo. These were the real game changers in immuno-oncology because they gave extraordinary successes, quite unexpected. And their efficacy is due to the fact that they allow reactivation of anti-tumor T lymphocytes. But not only, unfortunately, they also reactivate any lymphocytes that have been activated recently in the context of the therapy. And so these other T lymphocytes include also potentially autoreactive T lymphocytes, explaining why these immunostimulatory antibodies have very specific toxicities of an autoimmune kind.
Now, Professor Lucas, are there already drugs available for doctors and patients based on immunotherapy?
Yes, absolutely. There are drugs that have been approved by the FDA and by the EMA. And as far as I know, most of the drugs that are currently approved and used in everyday life, oncology, as you can say, are all directed against this CTLA-4 or PD-1, PD-L1 axis. So it's the anti-CTLA-4, anti-PD-1, anti-PD-L1 antibodies that are used either as monotherapies or in combination with one another, or yet in combination with other more conventional approaches in oncology like chemotherapy or radiotherapy.
This is a field where research is still going on and you're performing it. We need new breakthroughs. Where do you see the limitations for immunotherapies, or even where lie the challenges?
So I think that the main limitations are probably shown on this graph here and the following one. The main limitations are that currently not all patients and not all cancer types can benefit from currently available immunotherapies. So for example, you can see here the successes of the response rates, let's say, of metastatic melanoma patients to first, second or third generation immunotherapies, namely here anti-CTLA-4 antibodies, anti-PD-1 or anti-PD-L1 antibodies, or a combination of both. And you can see that there has been a huge progress by comparison to 15 or 20 years before, right? And now up to 50% of metastatic melanoma patients can respond very long term to these therapies. But still, the major challenge is that still half of the patients do not benefit from these therapies, even in metastatic melanoma, which responds to the therapies. The next slide shows that actually, if melanoma and lung cancer are cancer types that respond pretty well to these immunotherapies, this is not the case for all tumor types. There have been a lot of tumor types, histological types, that have shown very good responses to these therapies, but this shows that this is not the case yet for all tumor types, and in red, the circles in red indicate types of tumors that very rarely respond to these immunotherapies.
Another challenge that still lies ahead has to do with biomarkers. How can TCR sequencing help you in resolving that challenge?
Yes, so actually there are many biomarkers that are crucially needed, predictive biomarkers that would be predictive of responses to immunotherapy, biomarkers that can help monitor responses, that can help monitor toxicities or predict toxicities. The only biomarkers currently available are predictive biomarkers that are far from being perfect. For example, expression of PDL1 on the surface of tumor cells has been proposed as a biomarker for response to anti-PD1 or PDL1 therapy. Tumor mutational burden, which is the amount of mutation that can be found in the tumor of a patient, is associated, high tumor mutational burden seems to be associated with better responses to immunotherapies, and quite understandably, because a high tumor mutational burden can correspond to a higher load, a more important load of antigens on the surface of tumor cells. And then finally, there is a big potential probably for the use of TCR sequencing and the follow-up of the sequencing, the unique sequences of the T cell receptors that can be found on the anti-tumor T cells to follow responses to cancer immunotherapies, as these immunotherapies aim at amplifying these anti-tumor T cell responses.
Professor Sophie Lucas, thank you very much for sharing your insights with us and for introducing us into the field of immuno-oncology. We will see you later on in the Q&A session.
Okay, thank you.
And now, after the introduction into the field of immuno-oncology by Professor Sophie Lucas, let's take a closer look at the current therapeutic approaches and the innovation that is taking place in this field. We'll also talk about the remaining scientific and technical hurdles we still need to overcome in order to develop better treatments for the benefit of all cancer patients. I'm joined now by Dr. Eric Halioua. He's the CEO of the Belgo-French biotech company PDC-Line Pharma. His company is exploiting new, innovative scientific and technological approaches to expedite the research and the development continuum in the immuno-oncology space. And their aim is to bring new immunotherapeutics, such as cancer vaccines, to the patients. Eric Halioua, welcome. Can you please introduce PDC-Line Pharma and its activity to us?
Yes, thank you, Bjorn. So the PDC-Line Pharma is an immuno-oncology company developing a new class of cancer vaccine based on an off-the-shelf antigen-presenting cell line. In a nutshell, the company is a spin-off from the French Blood Bank, which is a large organization in France with the missions to collect blood from transfusions, but they have as well the specificity to have academic research lab associated. And one of his lab, led by Joël Plumas, the scientific founder of the company, was the first one to identify a very rare leukemia affecting the plasmacytoid dendritic cells. In the blood, you have the family of dendritic cells that are the competent cells to stimulate the immune system and to fight against viruses or cancer. And we have majority that are myeloid and a small fraction that are the plasmacytoid parts. And the patients had a leukemia affecting these small fractions. Based on the blood of these patients, Joël developed a purified cell line, amplified, and a master cell bank that we are still using today as a vaccinal platform. We are now a team of 31 people. We have, in terms of funding, raised close to 52 million euro. Our PDC line have three key advantages. It's very easy to expand. It's immortalized cell line. It's a leukemia cells that can grow, and I will show it later, in bioreactor. The origin of the cells being a plasmacytoid dendritic cells provide very interesting biological property in terms of capacity to expand the CD8 and to prime and expand the CD8 T cells. And last but not least, it's a versatile platform. We just have to load GMP peptide representative of tumor antigens, and we can have several of them loaded on the HLA present in the membrane of the cells, and therefore we can treat any kind of solid, potentially, or liquid tumors. We are clinical stage. The first feasibility academic study were conducted by Joël Plumas before the foundation of the company in melanoma, and PDC Line is focusing now on the phase 1/2 in non-small cell lung cancer on 64 patients.
Eric, you also have a very experienced management, haven't you?
Yes, indeed. We have a team of 5 people in the senior management team based in Belgium, in Liège, and in Grenoble, in France. People from GSK Vaccines, for the people based in Liège, and with experience as well in cell therapy biotechnology company. The scientific founder of the company, Joël full-time, the company, and myself, I've already co-founded several biotech, sold some of them to big pharma, and been involved in IPOs.
We all know that research and development in the biotechnology space is time-consuming and thus capital-intensive. You already have a first commercial success, haven't you?
That's right. In fact, we signed a first licensing deal with LG Chem. LG Chem is one of the divisions of LG, which is known, at least in Europe, for electronics, but LG Chem is the 10th chemical company in the world. The headquarters is based in South Korea, and we have licensed the rights for the development and the commercialization in Asia. They have an exclusive license in South Korea and options for the rest of Asia. The size of the deal is presented here. It's $123 million plus royalties on net sales. But at the top of the money, what is important is to have a partner, that knows the field, that know how to develop clinical trial in this area, that have access to the regulatory body peoples and the competent authorities, and that is very valuable for a company like us. We signed this deal in early 2019, before starting the clinical trial in non-small cell lung cancer.
Now let's dive a bit deeper into your scientific approach. How are you and your team developing those innovative cancer vaccines?
In fact, as I said earlier, we have this master cell bank with the cells from the blood of the patients, the PDC leukemia patients, that has been purified and amplified and fully characterized. And based on this master cell bank, we have developed several working cell banks to amplify, in fact, the quantity of the cells. And when we want to launch a manufacturing campaign, we just have to thaw the cells that are in liquid nitrogens and bag of the cells from the working cell bank. To seed single-use bioreactor, we have 1 liter, 10 liters bioreactors currently, and the cells grow in suspension in a synthetic medium without any calf serum or interleukin or cytokine. And after 9 to 10 days of cultures, we have billion and billion of cells. And at the harvest, we just have to split the cells in bags according to the number of peptide we want to load. We will load the peptide individually to avoid competitions because the peptides stick to the HLA present in the membrane of the cells. Then we pull the cells, irradiate to stop the proliferations. I remind you, it's a leukemia cells. Then we put in the vial of aseptic technology that are resistant to cryopreservations in clean rooms with a cryoprotectant. And we ship the cells in minus 80 degrees fridge in the hospitals. And like for the COVID-19 vaccine, when the patient is ready, they just have to thaw the cells in the hospitals and with a syringe take the cells and infuse to the patient's IV and subcut. And then the cells will go to the lymph node to prime naive T cells that will become effector and some of them memory later. And these effector T cells will go to the tumor microenvironment to kill the tumors.
You already told us that there's a clinical trial going on. How is this trial being conducted and what are the results so far?
Okay, let me start with the design of the non-small cell lung clinical trial. So we are enrolling currently 64 patients of this study on 4 different cohorts. The objective is to assess the safety, of course, first, but as well the immunological activity, the capacity of the vaccine to expand CD8 T cells in the blood of the patients, and the clinical response, which is measuring of tumor shrinkage and the durations of this tumor shrinkage. So we are testing 2 doses, low dose of vaccine and high dose of vaccine. First, with the vaccine alone in adjuvant setting. In these populations, the patient can have surgery to remove the tumor from the lung, and after, in general, chemotherapy adjuvant to remove any residual disease. They are infused, they have a washout period, and they are infused 6 times weekly with the vaccine. 6 patient low dose, 10 patient high dose. Second populations are metastatic patients. In this case, unfortunately, they cannot have surgery. There is tumor metastasis. And the standard of care after diagnosis is the pembrolizumab, checkpoint inhibitor. And here we are in associations after the diagnosis with the checkpoint inhibitor after the first infusions of with the first infusions of the pembrolizumab. And here again, we infuse 6 times weekly and we follow the patients. 6 patient low dose and 42 patient high dose. Why 42? Because it's in this last cohort we would like to observe statistically a significant difference. In terms of clinical response, we are going to follow 2 different clinical endpoints. What we call the overall response rate, which is a measure of the tumor shrinkage, pre and post-infusion of the vaccine, and the progression-free survival at 9 months, the number of patients that are stable or that response, so meaning that the tumor are shrinked. And we know, because it's published, for the pembrolizumab alone, the response in terms of overall response rate and PFS 9 months is 39% and 43%. Our objective is to increase this response by 15% and to get 54% for the overall response rate and 58% for the PFS 9 months. The result we get so far on the first cohort of patients, because we have completed the first one, not yet the high dose, what we call the B2, is out of the B1 patients where we can observe a clinical benefit, low dose on the 6 patients, we have already 4 partial response. We call partial response when we have more than 30% of tumor shrinkage in one of the time points. And we had 4 partial response out of the 6 patients. And in terms of PFS 9 months, we have as well 66.7 response, which is already higher than the 54 and 58% that was the target. Of course, on 6 patients, but it's definitely encouraging. When you look the tumor shrinkage, the amplitude of this tumor shrinkage, what you see here in this curve, as you can see here in green, the tumor shrinkage are not borderline. It's not close to 30%. Some of them are more than 85%. And we have a very interesting response on these 6 patients. And when you look the durations of the effect, here it's up to 12 months for the patient that has been up to 12 months yet, and the cutoff was end of May. As you see, the trend is very nice. We have on the term of duration of the effect for the responder, and that is definitely encouraging result we have pleased to have so far. In terms of immunological response now, and the idea is to look in the blood of the patients, pre and post vaccinations, what's happened, you know? And here we need to collect the blood. It's what you see in red in this slide. We have just before the infusions, we collect blood sample. 1 week post the 6 infusions of the vaccine, we have second blood sampling. 4 weeks post the 6 infusions, we have the third one, and the 4th one is 10 weeks post the 6 infusions. So we have 4 different time points, and the pembrolizumab, which is the standard of care in pink, is every 3 weeks. So let's see now the result of the first cohort of the patients. A1 low dose vaccine alone, A2 high dose vaccine alone, and B1 low dose with checkpoint inhibitor in metastatic patients. As you can see here, pre and post vaccinations, we have been able, for several of the antigen and the patients in their blood, post infusions, to see significant expansions of CD8 T cells, which is higher for the high dose than the low dose, and looks like there is a synergetic effect with the checkpoint inhibitor. And we will see better in the next slide. What is interesting as well to mention is post-infusions. We can, by FACS dextromer, measure the quantity of memory T cells. And the majority of these CD8 T cells are memory phenotype, so showing that the vaccine has been able to make the job, which is to transform naive T cell to memory T cells. We will analyze more deeply the response and use thanks to Seqalis technology. We will look for the sorted CD8 T cell for each of the antigens, the clonotype, and the quantity of this clonotype pre and post infusions to see what is more deeply the impact of our vaccine. What you see here is another way to see the result per cohort, and you have the fold increase per cohort. As you see, in red is the low dose vaccine alone. We have a range of 3. For the A2 cohort, high dose, you see the fold increase in average. The median is between 20 and 30. It's a bit lower for the B1, which is the low dose with the checkpoint inhibitor. So we definitely see immune response, which is increasing with the dose and fuse, and with anti-PD-1 led to higher levels. So we see the synergetic effect, which was in fact what were expected with our clinical trial. What is also interesting to see is the first signal of correlations we observe between the best overall response, the clinical response, and the immunological response in the B1 cohort patients. As you see here in this graph, for the progressive disease, the non-responder clinically, we don't observe any immunological activity. But for what we call responder, what are the partial response, or for the stable disease. In majority, 75% or 100%, we observe an immunological response. So this is the first signal that are definitely encouraging.
Well, Eric, these are very promising results. If we now look a bit further in future, what are your perspectives for the company for the next 2 to 3 years?
Definitely. The first objective is to complete the current clinical trial. We have this B2 cohort on 42 patients, which is still enrolling. We expect to have the final report of the full clinical study on the 64 patients beginning of Q2 2024. We should have an interim study report out of the 19% of the 42 patients in August 2023. And in 2024, we will prepare new clinical trials. That should be a randomized phase 2 clinical trial, and with the help of new funding, IPO, or deal with big pharmaceutical company.
Eric Halioua, thank you very much for sharing these very promising results. I wish you the best of luck in tackling the challenges on your path. Thank you for participating, and we'll see you again a bit later on in this session for the Q&A session.
Thank you. Thank you, Bjorn.
Now, in the third and last part of this seminar, we will exemplify the use of an innovative immune monitoring approach called quantitative TCR-seq within the day-to-day research and clinical trial activities of PDC-Line Pharma. And to do so, I'm joined by Dr. Joël Plumas, Chief Scientific Officer of PDC-Line Pharma, and Dr. Javier Carrasco, who is in charge of the research programs of the Department of Oncology of the Grand Hôpital de Charleroi in Belgium. Gentlemen, welcome. Could you please introduce yourself to our audience?
I'm Joël Plumas, co-founder and Chief Scientific Officer of PDC-Line Pharma, and I've been working on the technology for more than 20 years now.
Javier?
So I am Javier Carrasco. I am a medical oncologist and I work in Belgium, in the south of Brussels, in a clinical center that is the GSDC. Part of my working time is dedicated to clinical activities, and another part to research, clinical research, translational research, specifically in questions that turn around the way to monitorize and evaluate the immune system in patients treated with new immunotherapies.
Joël, let me start with you. Can you explain to us what the global rationale is and the objectives of using immunomonitoring in the development of your vaccines?
Yes, as mentioned by Eric today, we are developing a PDC-Line product and PDC-Lung01 product for targeting lung cancer patients. And this vaccine is made by a PDC-Line irradiated and loaded with peptide derived from tumor antigens expressed by the lung cancer patients we would like to treat. As a PDC-Line is an antigen-presenting cells, we expect to boost anti-tumor immunity in the blood of the patients. And by sampling at different time point pre and post-vaccination, we expect to catch these anti-tumor specific T cells in the blood of the patients by flow cytometry. And in this context, we decided to add a new methodology like TCR analysis to go deeper in the analysis and to try to detect more specifically the clonotype of antigen-specific T cells we are interested in.
So Javier, can you tell us what the basics of TCR sequencing are and the rationale for using it in immuno-oncology?
Yes, of course. Lymphocytes express a receptor that is called a TCR. This receptor allows the lymphocyte to recognize very specifically a precise antigen. When a naive lymphocyte, CD4 or CD8, recognizes antigen, he will be engaged in a differentiation program that includes an expansion at the clonal level. And it has been proposed that detecting this expansion could be interesting to monitorize or to have an information about ongoing adaptive T-cell immune response. But classical approaches, as for example flow cytometry, ELISpot, or immunohistochemistry, do not allow to catch this information. And T-cell receptor sequencing can be informative about this clonal expansion. The principle is to sequence the genes that encode for the TCR. These genes are very particular because they are engaged in a recombination process during the lymphocyte maturation, and each clonotype have a unique sequence. And we can say in a word that this sequence of this gene is a sort of ID card of the clonotype, of a T clonotype. So if we can sequence the genes, we will have a global information about the clonal composition of a T cell population. Of course, this information could be very interesting in a large number of fields, pathological, infectious pathology or autoimmune disease, but also in cancer. As Professor Lucas explained, immune system interact with cancer cell, and we know now that we can modulate this interaction. There is a number of treatments that are currently used in clinic that play on this modulation. For example, the anti-CTLA-4, anti-PD-L1. New treatments are under development, as for example the cancer vaccine that is developed by PDC-Line. But there is still some challenge. For example, determine which are the patients that will really benefit from this treatment, and also once we start the treatment, determine how to monitorize the efficacy of the treatment. And it has proposed by several teams in the world that TCR-seq could be an interesting tool to answer this challenge.
Now, sequencing highly variable genes such as TCR can be undoubtedly complex. What are the challenges involved?
Yes, indeed. It's not an easy job to sequence the T-cell receptor. As you mentioned, the genes are very variable, and there are some technical challenge. The first is the amplification bias. When you want to sequence a gene, you need to amplify the little fragment you want to sequence before you put in the sequencing machine. And during this amplification, you can introduce distortion on the proportion from one TCR compared to another one. The other problem, potential problem, is that there is this amplification, the enzyme that is used, that is a polymerase, can make some mistakes, and these mistakes will generate inaccuracy in the sequencing that can be interpreted as additional artifactual diversity. So these are two technical challenges, but there is more difficulties. When you look for something very diverse as a T-cell receptor repertoire, what you observe will strongly depend on the size of the sample you collect. This is easy to understand, but it's an important point that must take in consideration, especially for immunomonitoring application, where we want to compare blood, tumor samples, where the amount of T-cell is very variable between these different samples. So the size of the sample will have a strong impact on the observed result in immunomonitoring application.
So I understand that there are still a lot of hurdles to be overcome. What is the solution that Seqalis has developed in order to resolve these issues?
Well, we have been working on this to try to improve this challenge or these pitfalls since several years, and now we collaborate with Seqalis indeed. And the proposed solution is in fact based on a specific way to build the sequencing library. We add a little tag to each initial molecule before the amplification step. This little tag is a random sequence that allows to tag individually each molecule. And it's amplified with a global molecule, and once we obtain the read, we can compile all the reads that share the same tag. We call this a molecular barcode or unique molecular identifier. This compilation allows to correct, of course, the amplification bias, but also to decrease strongly the artifactual diversity. Maybe I can show you some data that illustrate the advantage of using this unique molecular identifier. In this experiment, you see, in the top of the slide, you see a comparison of the blood repertoire using an uncorrected approach. Each little dot corresponds to an individual clonotype, and the red dots correspond to clonotype on which we detect a significant difference between both replicates. You can see that there is an important bias that at least 21% of the measure are significantly different. In the second experiment, we test the approach in an artificial repertoire. In this case, it's not a biological sample, it's a mix of molecules. We know the sequence of these molecules, and we want to see at the end of the process if we identify the null sequence. And you can see in the slide that The gray dots that correspond to the expected sequence are detected, but there is a large amount of other sequences that are also detected that correspond to artifacts generated during the library construction. When we add the unique molecular identifier, you can see that we correct both problems. In the top, you see the correction of the amplification bias, and also you can see we correct almost completely the artifactual diversity. So, usage of unique molecular identifier allows to correct technical bias, but also there is another advantage. The unique molecular identifier allows to quantify absolutely the number of molecules that have been sequenced by counting the number of unique UMIs that we obtain among the reads. We have made some experiments to validate this quantification, and I can show you here one of these experiments. It's a simple experiment where we dilute an amount of molecules that contain TCR beta chain, and they were quantified by using a standard approach as qPCR, or quantitative PCR. This dilution curve was sequenced using the qTCR-seq approach, and we count the number of molecules that we recover, or that we sequence with this approach. And you can see in this figure that there is a very good correlation between both measures, indicating that the quantitative information corresponds to a reality.
Now, Joël, can you give us some more details on the practical implementation of immunomonitoring and how you used this in the clinical trials?
We use samples from patients, of course, and finally we have different time points where blood is sampling. And we have developed an immunomonitoring in 2 steps. The first is to collect the samples within 24 hours to purify the mononuclear cells and to freeze the cells for further analysis. When all the patients, all the samples coming from one patient are collected, we can proceed to the next step. And we received samples from different parts of Europe, Belgium, France, Netherlands, Germany, and also Poland. So during the second step, we thaw the cells, purify the CD8, and this is very interesting because we are looking specifically the CD8 T cells, and these cells are submitted to a multimer staining, meaning that we are using tools to detect specifically each antitumor specific T cells we are interested in. And finally, we submit these cells, labeled cells, for cell sorting to get exactly each expanded T cells we are interested in. And these cells are put in a special medium for TCR repertoire analysis at the end of experiment.
Does the qTCR-seq approach that has been developed by Seqalis also allow to use it on small samples?
Yes, as I just showed before, we can see that the technology allows to detect very small amount of molecules, as low as a few hundred. But with PDC-Line, we start to work to prove that the capacity to detect this small amount of molecules is also found if we work with cells, so very small amount of cells. So, and PDC-Line built a model, experimental model, where they dilute, well, it's short, decreasing amount of CD8 T cells, and as Joël explained, they extract RNA from this sorted T cell, and this RNA was sent to Seqalis in order to perform the TCR-seq. And you can see in the slide the result of this experiment that was performed in 2 independent experiments that revealed that we were able to detect TCR beta molecules in as low as 20 cells, maybe 10 cells in some cases. So this clearly shows that the technology is extremely sensitive, even with living cells sorted, for example, by FACS.
We talked about immunomonitoring in blood samples. Will you also be testing it in tumors?
Yes, and it is the idea. And the TCR repertoire analysis, is TCR-seq particularly, is a mean to demonstrate that the cells we can catch in the blood of the patient, so anti-tumor specific T cells, can reach the tumor by passing through the bloodstream. And it is particularly important to demonstrate that because we expect that all the immunity generated by the vaccines is able to kill the tumor cells where the tumor cells are, so in tumor bed.
Javier, is qTCR-seq applicable in tumor samples? What are your experiences in that field?
Well, it's clear it's probably one of the most difficult type of testing because collecting tumor samples is not easy. Sometimes the tumor sample is very small. And so we work also with PDC-Line team in order to validate if our approach could be applicable in the clinical trial expected. And this is the result of one illustrative data from one feasibility experiment. In this case, we include patients that have a lung cancer and that need a diagnostic test procedure with a biopsy of the lymph node in the mediastinum. And this biopsy is a fine needle aspiration, and material was collected and RNA was extracted from this material, biopsy. And we can see in the slide that we were able to perform the TCR-seq process and to detect a repertoire that is quite representative because you can see that we were able to detect more than 3,000 molecules in these samples. Another illustration to show that the approach can be applied in a tumor biopsy is this one. In this case, the patient included in this trial are rectal cancer patients that were treated with an experimental treatment combining radiotherapy and immunotherapy. And the trial planned to collect tumor biopsies at several time points during the treatment. And these biopsies were analyzed also by TCR-seq approaches. And you can see that we were able to characterize the T-cell repertoire at several time points. For example, one illustrative patient, we were able to track some clonotypes from one time point to another. And probably it's a good illustration of the advantage of to have a quantitative information because we can quantify exactly the amount of lymphocyte or molecule that we sequence, and this, of course, increases much more the power to have a significant comparison related to a quantification that was, in case this was limited only to a relative quantification.
Joël, what are the further perspectives of using the qTCR-seq approach within PDC-Line Pharma? What can be the potential impact of this technology on the immuno-oncology space?
I think the impact will be very important because we will have a better understanding of the immune response generated, the diversity of the response, the extent also of the response, and particularly when the TCR are not expressed on the cells, this molecular frequency of the TCR repertoire will help us to better follow all the clonotypes during the treatment. And at the end, we expect, as mentioned just before, the demonstration of the migration ability of anti-tumor specific T cells detected in the blood of the patient to migrate in the tumor where they will kill the tumor cells.
Javier Carrasco, to conclude this third part of the seminar, can you briefly wrap up the main advantages of the qTCR-seq approach and technology that you have developed here at Seqalis?
Yes, of course. So if we have to summarize, qTCR-seq approach is a molecular biology approach that allows to sequence T-cell receptor that use a unique molecular barcode to correct technical bias and also to bring an absolute quantification of the molecule sequence. This is a very important information very clearly in immunomonitoring application where we want to compare samples with very different amount of lymphocytes. As we have shown, the technology can be applied to very large type of samples, tumor samples, blood samples, but also sorted T cells and eventually culture cells.
Joël Plumas and Javier Carrasco, thank you very much for sharing all these details with us. I think we can go some more into detail now with the questions from our audience.
Thank you.
Thank you.
Welcome back, dear audience. Time now to dive deep into the subjects that you would like to discuss. And let me reintroduce our different panelists. Professor Sophie Lucas of the Belgian de Duve Institute, Dr. Halioua, CEO of PDC-Line Pharma, Dr. Joël Plumas, CSO of PDC-Line Pharma, and Dr. Javier Carrasco, a clinical oncologist from the Grand Hôpital de Charleroi. If you have one question that you would like to ask to one of our panelists or multiple panelists, you can easily send them through the chat function on your screen. Professor Lucas, let me start with a question for you on the anti-GARP therapeutic antibodies. What differentiates these antibody candidates currently in phase 1 clinical trials from other available cancer immunotherapies?
Well, thank you for the question. I think there are several things that distinguish these antibodies, and probably mainly three. The first is the cells that they target. So these antibodies would target Tregs rather than the effector T cells, right? And so they would target an independent mechanism from that is usually being targeted by the anti-PD-1 or the anti-CTLA-4 antibody. And so it could increase the efficacy of the already existing immunotherapy. So that is the major difference, that independent pathway. The second thing that makes them really different is that while targeting Tregs, they target the immunosuppressive function of the Tregs rather than trying to deplete these cells. So there are other approaches that are aiming at decreasing the suppression by Tregs, but none of them have reached the clinics yet, and most of them attempt to deplete these cells. But without Tregs, you have very severe autoimmunity. So we hope that by transiently inhibiting the function of the Tregs rather than completely eradicating these cells might be less toxic than what is currently being tested. And then finally, a third thing is that while trying to target Tregs rather than effector T cells, to target the function of Tregs rather than trying to eliminate them also, we are trying to target only one of their immunosuppressive function, which implies the production of TGF-beta. And TGF-beta is, of course, being also targeted by other approaches. It's a cytokine that other people try to target also, but there again, by using the anti-GARP antibodies, we are aiming at reducing the production of TGF-beta activity that emanates from a single cellular source. And TGF-beta has always been a very, very difficult target because it exerts a lot of different effects, a lot of pleiotropic effects. And so here we have a means perhaps to target this very interesting cytokine, but not all its activity, only that emanating from Tregs. So that is how I would explain what makes anti-GARP antibodies different from the other approaches that are available out there.
Do you already have an idea for which types of cancer these anti-GARP antibodies could be effective?
Well, actually, they are now being tested in a first-in-human trial, in a phase 1 trial, sponsored by a pharmaceutical industry. And so they are being tested in combination with anti-PD-1 in different types of solid tumors. And the different types of solid tumors that are currently being tested include urothelial cancer, non-small cell lung carcinoma, hepatocarcinoma, head and neck cancer, pancreatic cancer was also tested.
Okay, thank you very much. Joël Plumas, a question for you. What is the limit of quantification of the immunomonitory assay that you have developed?
It is a good question because we still need to have a limit of quantification to define an expansion of antigen-specific T cells. And in this case, decreasing the limit of quantification is not an easy task because currently what the limit we reach with the current immunotherapy assay we develop is 0.003%. So it is very low, and by looking at this type of limit of quantification, we are able to define very precisely and very with some confidence a twofold change that it is really meaningful when we look at the expansion of antigen-specific T cells in the blood of the patient.
Have you had the opportunity to detect expansion of antigen-specific T cells, to sort them and to analyze their TCR repertoire?
Yes, we were very happy to already have some immuno results in our current clinical trial, and specifically with low dose of the product, but also with high dose of the product, and again also in combination with anti-PD-1. And we have sorted different antigen-specific T cells against different specificity, MHA3, Survivin, and NY-ESO-1. And we succeed to make RNA-seq analysis of these antigen-specific T cells, and we are ongoing analyzing this phenotype and the diversity of the repertoire, and how we can use this information to follow all the antigen-specific T cells all along the vaccination schedule in the different samples we have.
Thank you, Joël, Plumas. Dear audience, if you still have a question for one of our panelists, do not hesitate to send it to us. It's very easy. You can use the chat box next to this video screen. And now a question for Javier Carrasco. TCR-seq is of increasing interest in the field of immuno-oncology. It has been proposed as a tool for monitoring adaptive T-cell responses. What is your perspective on its use as a biomarker to predict and to monitor the efficacy of immunomodulatory therapies currently used in the clinic?
Yes, this indeed is a very relevant question, I think, because identification of predictive biomarker for immunomodulating treatment is still a pending question. And well, it has been proposed that T-cell receptor sequencing as a tool to analyze adaptive T-cell response, and we know that T-cell response play an important role when we use immunomodulating treatment, but it has been proposed that this tool could be used to predict or to follow the efficacy of immunomodulating treatment. For now, there is no clear data. I will say there is some publication that suggests that the repertoire in the blood could be, some specific pattern of the repertoire in the blood could be linked with the efficacy of the treatment, also in the tumor. But there is very, I would say, divergent and contradictory data. Probably one of the difficulty or challenge is that when we use T-cell receptors, discussed in the presentation, T-cell receptor sequencing depends very strongly from of the size of the sample that is analyzed. And of course, if we use this technology in several types of tumors, and we know that tumors are very variably infiltrated with anti-tumor T-cell, we will have very variable result depending on this infiltration. And this is why I think that the usage of quantitative information, also technology that have allowed to quantify absolutely the number of molecules that is sequenced, is potentially useful to improve the accuracy of the test and potentially use as a biomarker in the clinical approaches.
Another question for you. qTCR-seq approach looks very interesting as a standalone approach, but it has already been associated with technologies such as spatial biology and 3D transcriptomics. Could you explain how and what was the scientific outcome of such investigations so far?
Yes, this also an interesting question, because when you do T-cell receptor sequencing, you have information about, I will say, just a piece of the global puzzle of the adaptive T-cell response. You don't have information, for example, about the functionality. You don't have information about the specificity of the clonotypes you identify. So this is why it is useful to combine this approach, maybe with other approaches. Some groups in the world use single-cell approaches. Other groups of PDC-Line use dextramer sorting in order to capture information about the specificity. And as you mentioned, now there is a new approach that is called the spatial transcriptomic that allows to have a spatial view about the gene expression in, for example, a tumor. And with the group of Seqalis and also our research lab, we have developed an approach to identify individual clonotypes in the tissue. And we are now able to detect these clonotypes and analyze their distribution in the tumor, and also associate this each individual clonotype with specific functional genes, effector genes or differentiation genes. We presented a poster recently in the ESMO Congress, ESMO or immuno-oncology in December in Geneva, about this approach.
A question which I think probably would be interesting to hear multiple points of view, maybe also from Eric Halioua in a moment. What are the most promising immunotherapy targets with no product on the market yet? Maybe first your opinion, Professor Lucas?
Yes, well, that's always a question to which it is difficult to answer without any bias, because particularly when you're asking that to researchers that are implied in the development of novel immunotherapeutics. But that being said, I would say that actually the most important inhibitory receptors on T cells have already been targeted, right, with anti-PD-1. So obviously, the most promising targets will be on other cell types. And that is why I am so much convinced that targeting Tregs will be extremely important, because then you would be able to combine that with the currently available approaches, hoping to increase efficacy. Other cell types could, of course, be targeted, like MDSCs and like cancer-associated fibroblasts, but there we are very, very far from having anything that is testable even in the clinics. But then also, I think that the way to go in the future is certainly to increase the specificity of the anti-tumor responses that can be induced with the available therapies or the next generation therapy. And I think that their vaccines have a very, very interesting potential, particularly vaccines that use the so-called tumor-specific antigens. And so I would really insist on the fact that not all vaccines are interesting. Those vaccines that use antigens that are actually completely specific of the tumor cells, such as those that were discovered by Thierry Bohn and his colleagues, and I see that you are using at PDC vaccines, such as the MAGE-3 vaccines or MAGE-3 peptides that are particularly interesting because they are also shared by many other patients. So using vaccines with tumor-specific antigens would allow to increase the specificity of the targeting against the tumor, increase perhaps the specificity without increasing the toxicity, or even allow to reduce the doses of the anti-PD-1 antibodies, for example, that are being used to reduce the autoimmune toxicity that you can see with these.
Eric Halioua, what do you consider to be most promising immunotherapy targets not on the market yet with a product?
Not on the market yet. So you know that today there's a lot of approach that are in development. And as said by Professor Lucas, I would agree with his analysis. Some of them are in a really early stage. But if I try to summarize, there is target related to the immunity, amplification of the immune system, which is where the vaccine is playing. You have over target related to the tumor microenvironment, you know, where we try to elucidate why the tumor can defend himself or to avoid whatever invasions or activations of the CD8 T cells. And here is a lot of different approach or target related to whatever the T cells or the dendritic cells. So macrophage as well is one area of interest by some organizations, different kind of macrophages and T cells as well, Treg or different category of effector or memory T cells. So, but once again, I think today, as has been explained, the game is the game of combinations. Therapy in immuno-oncology with the checkpoint inhibitor, we know that we can at least address the mechanism of defense of the tumor, that can inhibit the T cells. And that opened the door definitely by breaking these mechanisms, the ability of expanding cytotoxic CD8 T cell through vaccines and to have enough mass of CD8 T cell infiltrating the tumor to make the job, which is to kill the tumors. So, and I strongly believe that these kind of combinations will be one of the approach with vaccine, checkpoint, vaccine, and other things to be able to answer to the different need and different patient populations. Because according to the indications, according to the level of development of the disease, you could adjust, once again, the therapeutic arsenal.
Javier Carrasco, finally, can we have your point of view as well?
Yeah, of course, it's a very large question. So clearly, there is in the clinic, we see, well, when I say clinic, in clinical research, we see several strategies. One of the strategy is, as Eric mentioned, is combination, and not only immunotherapies with immunotherapy, but also chemotherapy with immunotherapy, radiotherapy with immunotherapy. There is a large number of combinations, targeted therapies with immunotherapy, and we don't know, at least for the moment, which is the winner of this type of combination. There is also new immunomodulating treatment that are really in the pipeline. I think about, for example, anti-LAG3. That all seems very promising, very interesting result in melanoma, anti-TIGIT antibody. But as Sophie, Dr. Lucas mentioned, there is also other strategies like vaccination. And I agree that specific tumor antigen are crucial, but a group of antigen that is called neoantigen seems to be very interesting. And we see now in the clinic new strategies of vaccination that use a personalized vaccine. So that means that we build a vaccine for a patient and we build this vaccine based on the expected neoantigen that are expressed or present by the tumor of the patient. We participate now in a trial that evaluate this strategy, for example, in localized rectal cancer with high risk of relapse, and the trial evaluates usage of this type of vaccine after the surgery in order to prevent the relapse of the disease. We will see in the future. I am really I look forward to see this result. I think it could be promising approaches.
We can clearly see that you are excited about that. I have a question up for Dr. Joël Plumas. How do you select specific CD8 plus T cells? And by this selection, can you guarantee that we do not lose some information about unselected T cells?
In fact, from peripheral blood mononuclear cells, we are selecting CD8 T cells, the whole population of selecting T cells, by doing negative selection. This means that we do not exclude any CD8 T cells. So we are looking for all the population, and this whole population is submitted to the multimer staining and then the sorting cells. And by doing that also, we still keep the whole population to look at specifically with the TCR sequence of specific clones, how this specific clone is represented in the whole population of different of CD8 in different time points. So there is no bias, I think, by doing experiment as we do.
Javier Carrasco, I saw you nod. Would you like to add something?
No, no. I think it's just a comment, but I think that the approach that is described by Joël is really interesting. They, of course, capture information on specific T cells that are labeled by dextramer, but also in the negative part, not labeled. And clearly, the T cell receptor sequencing allowed to localize each clonotype in, well, of course, inside a population, a specific dextramer population. You can see the composition at the clonal level, and then follow this clonotype in the global CD8 and also potentially in other compartments, like, for example, the tumor, or this is a more global potential application if the sample are available, of course.
And then maybe another question for both you, Javier Carrasco, and maybe also for Joël Plumas. Could FACS assays or HLA tetramer combinatorial coding assays be used with TCR-seq? And in which phase or study or patient case would you use such an approach? Javier, maybe you can start.
Yes, well, this maybe Joel is in better place than me because I have a strong experience in dextramer, and this type of labeling. But I think this is an approach that can be combined also with. So it's an approach that allows to label some specific T cell based on the specificity. The approach allows to detect a large number of specificity, and as I mentioned, it is interesting to combine this type of technologies with T cell receptor sequencing because the technology gives the information about the specificity, and the TCR-seq allow the tracking of each specific clonotype.
Joël Plumas?
Yes, I think it is feasible. The sole limit of this technology is the number of cells you can select by each specificity. Doing a TCR sequencing of expanded T cells needs to get enough cells to be sure that the T cell repertoire you are looking at in small number of cells is representative of the population. So if you have a great expansion of antigen-specific T cells for many specificity, I think it is easy to combine the approach. But in case of a low expansion, and it's why I discuss about the limit of quantification, and the limit of detection, I think less maybe 0.01%, it is very hard to combine this approach, this type of approaches.
Well, thank you very much. We have treated more or less every question that was asked, so we are getting to the end of today's live panel entitled Immunotherapies, New Perspectives for the Treatment of Cancer Patients. If you require further information, dear viewers, do not hesitate to contact us via the website seqalis.com. On behalf of the company Seqalis, I would like to thank you, dear audience, for your participation and for sending us your questions today. And we would also like to extend our appreciation to the four panelists of today, Professor Sophie Lucas, Dr. Halioua, Dr. Joël Plumas, and Dr. Javier Carrasco. Bye-bye for now, and enjoy the rest of your evening.